Recording in progress.
Okay, we will get started. So welcome everyone to the Klein Scientific webinar series celebrating live cell imaging. We're here today with Tianjiao Dai from Luminicell to discuss innovative fluorescent probes for live cell imaging. So before we start, I just wanted to briefly cover the scope of Klein Scientific. We're a distributor in Australia. We focus on optical microscopy. We have a whole range of suppliers from microscopes, from Nexscope, pressed optics, confocal and super resolution systems. Now, recently announced in August, vivo microscopes and motion control. And we also do reagents from Luminicell, Spirochrome, and others. So we've started this webinar series to really celebrate live cell imaging in the spring in the Southern Hemisphere. So we're coming out of winter, but we can't quite wait. We are at the end of August, and we wanted to highlight all of our suppliers that have something to do with live cell imaging, which, to be honest, is most of them. So we are having webinars from Ocolabs, from Pressed Optics, from Spirochrome, Zaba, and many more. And we want to really highlight what our suppliers offer in live cell imaging. And we want to really communicate to researchers the tips and tricks and the new products that are out there. As I visit customers and visit imaging facilities, it's fantastic to see the cool research that everyone's doing. But I think it's really striking how many new developments there have been, especially in the last 10 years, around live cell imaging focused products and applications. So we wanted to really highlight some of those. So I'll leave this screen. That's all from me for the moment. At the end, you'll have some information and a short survey, but please get in contact with me at ben@kleinaustralia.com.au for quotes and more information. And our next webinar will be imaging dishes and plates with Cellviz and Matrogen. And Matrogen is a company that develops soft hydrogels coating dishes and plates for live cell imaging and cell culturing. That's on September the 12th. So I'll hand over to Tianjiao now and take it away. I think you just muted Tianjiao.
Can see my screen, right?
Perfect.
Okay, great. Okay, let's start now. Okay, good morning,
Fantastic.
everyone. Thank you for joining our webinar today. And I'd like to extend a special thanks to Ben and Klein Scientific for hosting and supporting this session. It's my great pleasure to present on behalf of Luminicell and share more about topic long-term live cell imaging. Before we dive into the main topic, let me briefly introduce our company. Luminicell was founded in 2014 by Professor Liu Bing from National University of Singapore. And in 2022, Luminicell became a wholly owned subsidiary of Nanolumi, which is also a Singapore-based company specializing in advanced luminescent materials. In Nanolumi, we have two different verticals with the Grand Rail focusing on product authentication and the Luminicell dedicated to advanced bioimaging. And now let's get into the main topic, bioimaging. So why do we need long-term imaging? We know that in biomedical research, especially the end-to-end translational research, there is always a critical need to study the occurrence and progression of the disease, and also other biological process over time. For example, if you are a cancer researcher, you may want to observe the occurrence of primary tumor and also the whole metastatic process. And similarly, stem cell researchers often need to track the distribution of the transplanted stem cells and also the fate and viability of the stem cells over an extended period. And the similar problem also exists in other biomedical research fields, including the immunotherapy and organoid spheroid research and others. And so here the challenge becomes clear. How can we study a process if we can't see it? Fluorescent labeling is the most common method used for visualization, and there are several types of fluorescent label available today. Among all these technologies, fluorescent proteins such as GFP and RFP are popular for long-term tracking, but they require laborious preparation in advance, often taking times, which often takes weeks, and involving genetic manipulation of the cells, which makes them inappropriate for some cases. The second type, organic dyes, particularly the small molecular organic dyes, including the cyanine-based dyes and the fluorescein or rhodamine-based dyes, tend to suffer from photobleaching. In other words, the signal is weakened easily, making them less ideal for long-term tracking. And the third type comes to quantum dots, which were recognized with the Nobel Prize in Chemistry last year, are considered as one of the brightest materials in the market. However, they have a major drawback. The heavy metal core of quantum dots exhibit toxicity to cells, leading to cell death, and thus they are not the best choice for live cell imaging. To meet all the need for long-term bioimaging, Luminicell proposed a new solution, nanoparticle-based fluorescent trackers. These nanoparticles are composed of 3 different layers, and each representing a unique innovation. Starting from the innermost layer, we encapsulate the AIE fluorescent dyes in the core. AIE, in short of aggregation-induced emission, is a phenomenon first discovered by Professor Tang Benzhong from Hong Kong University of Science and Technology in 2001. And what's the specialty of AIE dyes versus common dyes? Let's make a comparison together. So traditional small molecular dyes usually have a planar structure, and they are highly emissive in solution, but lose signal in aggregates due to peptide stacking, termed as aggregation cost quenching, ACQ in short. And on the contrary, typical AIE molecules are non-emissive in solution, but become highly emissive in aggregate due to the restriction of intramolecular motion. And these images show that while adding water, emission is weakened for ACQ dyes, but intensified for AIE dyes. And here's the video. And this property actually makes AIE dyes ideal for encapsulation in the nanoparticle core. Now the second layer. The second layer of our nanoparticle involves encapsulation technology. We have developed a patented platform to encapsulate AIE fluorescent dyes with amphiphilic lipid polymer matrix into nanoparticles. This technology allows us to produce the nanoparticles uniformly and reproducibly, with the size tunable between 20 and 90 nanometers. Our encapsulation technology platform enables the challenging materials and also hydrophobic materials to be suitable for in vivo applications. Moving on, the third layer involves surface functionalization. Beyond encapsulation, we can further functionalize the nanoparticle surface with different targeting and functional groups. We have our proprietary methods for conjugating different biomolecules, such as aptamers, peptides, and also antibodies, which enable us to control the cellular uptake efficiency and to achieve selective targeting towards tumors or specific organs. So this sophisticated design integrates all the necessary benefits into one system to satisfy the needs of bioimaging. With all these designs, we have developed our products, Cellaris, previously known as Luminicell Tracker Cell Labeling Kit. Cellaris is optimized for cell uptake and supports a wide range of cell applications. Our flagship products are 540 and 670 with green and red emission. And beyond that, we've also expanded our product range. We now have the blue and cyan products that can meet the multiplexing needs, as well as the near-infrared products, including the NIR1 and 2 for applications requiring deeper penetration depths, pushing the boundaries of limit even forward. We foresee that near-infrared imaging will become increasingly important, with more applications emerging in the near future. And at the bottom of this page, the listed are our distribution partners in global. And Klein Scientific is our strategic partner covering Australia and New Zealand. Apart from Cellaris, we also have vascular trackers. They are designed with the minimum cell uptake, and there's no leakage from blood vessels. From this slide, it starts to show the absorption and emission spectra of our 6 products. The dotted lines represent the absorption spectra, and the solid lines represent emission spectra. This page shows the blue and cyan, and green and red here, and also NIR1 and 2. So from all the spectra, we can tell that AIE dyes provide unique spectra with a broad emission and a large Stokes shift, which means that the absorption peak and emission peak are far away from each other, so that AIE dyes are less prone to quenching. Also, we have validated the 2-photon absorption, and here's the example of 2-photon absorption spectrum of Cellaris 670. Compared with another emissive dye, Evans blue, we can tell that Cellaris 670 has a much higher sensitivity for 2-photon excitation, resulting in an outstanding signal-to-noise ratio and better imaging depth. After introducing the products, let's talk about the performance validation. First, we validated the brightness of our products by comparing them with quantum dots, as they're one of the brightest materials on the market. As shown in the figures, Cellaris emits 40 times more photons than the quantum dots, capturing 100 seconds, demonstrating its ultra-high brightness. Next, we assess the physical and photo stability of Cellaris. The results show that Cellaris has higher physical stability than quantum dots and greater photo stability than FITC, which is a small molecular dye. Okay, so how about cell labeling efficiency? We have tested Cellaris in different cell lines. The confocal images show that Cellaris, as a non-transfective reporter, achieves rapid cell labeling with a high labeling rate across different cell lines. Moving on, we also validated the capability of Cellaris for cell lineage tracing purpose. From the flow cytometry data and the confocal images, we can conclude that Cellaris can trace the cells in vitro for more than 10 generations. Compared to quantum dots, Cellaris offers 3 times longer tracing duration and 10 times higher brightness. We know that safety is paramount in biological applications. Our data here shows no significant body weight changes in mice after treated with Cellaris, and no lesions or damages caused to organs, indicating negligible biointerference. We also have in vitro tests showing that after incubation with Cellaris for 48 hours, the cell viability remains above 95%, indicating limited toxicity. To summarize, Cellaris and vascular trackers offers a 10-fold brighter signal and a 3 times longer signal longevity for long-term tracking, and excellent biocompatibility with negligible biointerference. In one sentence, Cellaris and vascular trackers are designed to be the best-in-class solutions for end-to-end translational biomedical research. Now I've shared the design principles and product performance validation. I'm sure you're curious about the applications our products can be used for and what advances can be made in translational research with Luminicell products. We have categorized the applications into 3 main different fields. First, vascular imaging, where our vascular trackers offer high brightness and outstanding photostability, and most importantly, the compatibility with multiphoton microscopy, enabling the intravital imaging and mapping of vasculature. Second, visualization of 2D and 3D in vitro models, where Cellaris provide rapid and efficient cell labeling and good cell retention. And importantly, there's no crosstalk between different cells, means there's no leakage from the healthy cells, making them ideal for studying immune cells' interactions with cancer cells. And finally, vivo long-term cell tracking. The benefits adding here are the excellent biocompatibility and also the compatibility with in vivo imaging systems. Cellaris are perfect for cell therapy applications, allowing researchers to track the cell distribution, viability, and the fate post-transplantation. Let's take a closer look at these applications, starting with vascular mapping and leakage detection. We have used the multiphoton microscope to observe the real-time blood flow in the vessels intravitally and to create clear 3D reconstruction of mouse brain vasculatures as well. The high resolution and signal-to-noise ratio of the images showcase the capabilities of our red and green vascular trackers. And this slide highlights ex vivo organ fluorescence imaging using vascular tracker 670, which was injected prior to organ harvesting. Kidney image and video on the left and brain on the right reveals clear vascular details, emphasizing the outstanding brightness and photostability that our trackers provide, even in the complex tissues. And this slide presents in vivo whole mouse imaging using vascular tracker 1010 in near-R2 optical window. Performed 1 hour after intravenous injection of vascular tracker 1010, the images showcase the exceptional details captured with near-R2 imaging. Our vascular trackers are also effective for monitoring vascular leakage. We know that malaria infection causes hemorrhage, and we want to detect blood hemorrhage size. So in this study, vascular tracker has been applied to stain the blood vessels of the infected and non-infected mice. The images show that our trackers identified hemorrhage sites that were undetectable with conventional dyes like Evans blue, demonstrating their capability for monitoring the integrity of blood vessels and the potential for clear and early identification of the blood vessel damage caused by infection or inflammation. Now we move on to talk about Luminicell solutions for visualizing in vitro models. We know that there is no leakage of Cellaris from healthy cells, so it can be used for cell-cell interaction study. For instance, in the study where MDAMB157 cells were stained with Cellaris 670 and adiponectin expressing regulatory T cells with green dye, we observed that the red cancer cells engulfed the green Treg cells, showing a cell-in-cell phenomenon, which led to cancer cell death. And this labeling approach could be meaningful for studying how immune cells react to cancer cells and how they function. Organoid and spheroid research is gaining more and more traction, especially after the FDA's recent decision to exonerate the requirements for animal studies before clinical trials. So organoids and spheroids are now recognized as effective tools to mimic the human body environment and predict physiological response. Monitoring the organoid and spheroid formation process and studying the interaction or competition between different cell types has become increasingly meaningful. To address it, we proposed the bottom-up labeling approach. In collaboration with our German partners, Cantalab and Perspective Instruments, Cellaris was used to label the cells before forming the spheroid, and afterwards, the entire process of spheroid formation is monitored under microscope. The standard images on the left are the spheroid harvested on day 7, compared to the post-fixation stained Hochschild and Faladin, which only show signal in peripheral areas. The bottom-up labeled Cellaris emits more uniform signal through all the different layers of spheroid, demonstrating the excellent brightness of Cellaris and also the superiority of bottom-up labeling. On the right, we also have the spheroid that was continuously irradiated with a 2-photon laser for 30 minutes, during which the signal of Cellaris remains bright, proving its outstanding photostability. And the next slide shows application of vascular tracker in visualizing the vessel model in microfluidic device. On the left, the wild-type model shows an intact vessel structure with negligible vascular tracker signal leakage. And on the right, the loss of function model have more leakage size, as indicated by the yellow signal of vascular tracker and pointed out by the white arrows, demonstrating the compromised integrity of vascular tracker of the vasculature compared to the wild type. And this application is actually from our Australian partner, Dr. Anne and her team at University of Queensland. To continue, Cellaris have been widely used to track the in vivo distribution of the transplanted cells. This slide shows the schematic process of in vitro cell labeling and in vivo monitoring. Cellaris offers a simpler workflow compared to the gene transfection method, with less labor and time required. This first application case is long-term tracking of adipose tissue-derived stem cells. In this application, long-term tracking using Cellaris has been studied both in vitro and in vivo. From the in vitro confocal data on the left, we can observe that Cellaris has much higher brightness and a much longer signal longevity than the other commercial dyes. And in the in vivo data, it also tells that after Cellaris labeling and the transplantation in vivo, the signal of the stem cells can still be detected on day 42 post-transplantation, proving the brightness and the longevity of Cellaris. Another application is long-term tracking of tumor cells and also monitoring of tumor formation process. In this project, Cellaris 810, which is an NR1 emissive product, has been used to label the tumor cells in vitro, and then the labeled cells were subcutaneously transplanted into the mice. And the Q-tracker was also used as a control for the whole experiment. From IVIS images on the left, we can tell that after day 31 of the transplantation, the signal of Cellaris can still be clearly detected, while for Q-tracker labeled cells, the signal can only be seen on day 5 or 7. This quantitative data figure on the right side verifies the high brightness and much longer signal longevity achieved by Cellaris. In this study, Cellaris was used to track the stem cell-derived neurons in vivo. Salaris has been proven with negligible toxicity caused to the derived neurons. As for in vivo application, the signal of Salaris labeled neurons can still be detected even after 1 month, showing on the right images. And without the listed benefits at the bottom, the long-term tracking could not be achieved. And there are more applications for the in vivo biodistribution. In these two studies, xenogenic cells have been applied to activate the immune system of the tumor-bearing mice to induce immunotherapy. So briefly, xenogenic cells were stained with Salaris and injected into the mice situ. From the images, we can observe the signal of Salaris till day 21. And to confirm, the researchers also applied PCR to track the location and longevity of xenogenic cells post-transplantation. Salaris signals are well correlated with the PCR data, proving that the capability of Salaris for long-term in vivo biodistribution study. Here are two more case studies. On the left, Salaris was used to label and track the biodistribution of the injected mesenchymal stem cells, and the organs were harvested after 2 weeks of transplantation. And from those ex vivo organ images, the transplanted MSCs have a significant higher signal in the lungs and indicating that the MSCs were homing and accumulated in lungs. On the right, the researchers make use of the long-term tracking ability of Salaris to label MSCs and study the different effect of reduced and oxidized HMGB1 on the tumor infiltration behavior of MSCs. Last but not least, in our strategic plan, we are also targeting the application of fluorescent-guided surgery in the long term. And this is our preliminary proof points. The mice with peritoneal carcinoma were injected with our NIR fluorescent solutions via their tail veins. And before surgery, the tumors could be clearly identified with a high signal-to-noise ratio. And also, the fluorescent probes could also enable the surgeon to remove smaller nodules, which were otherwise undetectable when performing the surgery without guidance. And these presented data are just as examples of our proven use cases, but we're not limited to this. And here's the summary of the benefits of our products. So briefly, we have super high brightness and we have signal longevity, opens possibilities to perform long-term life cell tracking. And also, the encapsulation matrix enables the challenging materials to be delivered in vivo in a biocompatible way. And more importantly, our method does not contain any genetic modification. Therefore, there's no affection on the cell's normal behaviors. And according to different users' feedback, our protocol are easy to use and the workflow is quite simple. And also, our highly tunable platform technology allows our fast turnaround for customized solutions. And at the last, as a reminder that we have a full spectrum solution of Salaris for various cell-related research applications. And we also have vascular trackers used for mapping vasculature and detecting the vascular leakage size, also available in different colors. Beyond that, we also offer customized conjugated products and conjugation services as well, aiming to meet researchers' different needs on fluorescent labeling of biomolecules. And here's the summary of our partners and customers in global. I hope I didn't talk too much and you are still interested to learn more about us. And here's our official website where you can find our product information, news release, and our technical and application notes as well. Also, we'd like to invite you to follow us on LinkedIn and X to read our weekly sharing regarding fluorescent imaging and to see our events updates. That's almost the end of sharing. Thank you for listening, and I'll just transfer it to Ben.
Fantastic. Thank you so much for the interesting presentation, Tianjiao. So we might just keep you connected. And if anybody has any questions, please put those in your chat Q&A tab, and we can answer those. And in the meantime, I'd just like to highlight that our next webinar will be advertised online and on LinkedIn, and it'll be on September the 12th at 10 a.m. Melbourne time. We will be presenting with CellViz and Matrigen and talking about how high quality imaging plates can improve your imaging, and from Matrigen, how soft materials can help cell growth and improve your live cell culture behavior with that. So that should be a really interesting one, because it'll be talking about the physics of cell growth and how you can tune your cells and your plates for improved cell growth. So please look out for that then. So I don't think we have any questions open at the moment. So if anyone has anything further to ask, please get in touch with me or Luminicell, and we'll be happy to answer your questions. And I hope to see you at our next webinar in September. Thank you so much, everyone. We'll be in touch soon.